Thanks, Hyun. Thanks for really wonderful introduction. I'm very thankful to AKN and the organizer for inviting me today. And I'm very happy to share my recent work about the new direction from our lab, which is as regulation of headache pain response by the circadian clock. So just dive in. So the circadian clock is existing in all the living organism on Earth because of the Earth rotation around the sun create very two distinct environmental change, which is day and night. And living organisms try to adjust to these two different stressful environment, keep changing environment, and they develop this endogenous molecular clock to accommodate this change and then maximize their physiological well-being and survival. So before human actually have science or anything from before even Jesus Christ born like a 8 AD. There is a description in a lot of books that there is a circadian rhythm in plant. And here I call this as a sun dance, that this is a sunflower and then following the sun move. And I'm gonna just fast forward and then you were gonna go down. And then during the night, the leaf is going down. But interestingly, before the sunrise, this leaf is ready to receive the sun and then ready to do photosynthesis as soon as the sunrise, and then it goes the cycle. So this is really strictly, the plant move is strictly regulated by circadian clock. And in 18th century, de Marine, he is the first one actually who described the circadian rhythm in the science literature by observing the mimosa's leaf movement. So it opened during the daytime and then it goes down and close the leaf. And then he did the experiment in the constant dark condition. This leaf movement exists. So without external cue, this endogenous built-in clock was able to move the mimosa's leaf movement. So plant circadian biology was like very early described. And then human circadian rhythm was described only 1938, that people know that they have rhythm, but this is almost first experiment trying to measure human sleep rhythm. And then Nathan and then his student Bruce, they went to cave to create the constant darkness environment and then record their sleep cycle. So probably, I guess this is a student and then this is a professor recording, but back then, maybe this is professor or this is student, I don't know by picture. But from this study, for 6 weeks in isolated compartment, which is constant dark condition, they record that human sleep and cycle or human circadian rhythm exists, and then human cycle is actually longer than 24 hours, close to 25 hours, according to their recording. And then later, people did another experiment. A lot of people did this experiment in the constant condition. This is constant light condition. So they do entrainment in LD cycle and then do the constant light condition and still showing the little bit of longer period. So plotted by 12 hours. So if it's longer than 12 hours period, it looks like they're getting to bed a little bit late, but this is because of their endogenous clock is longer than 24 hours. And then when you go back to the LD condition, light, dark, controlled environment, they can reset. And then when you release them to the constant light condition, they can free run again. So this sleep recording really manifests the circadian clock is existing and regulating human behavior of sleep. And the interesting thing is, like we wonder when people really develop circadian rhythm, and then probably you guys know that when baby born, they don't have circadian rhythm. And about exactly around 100 days, they start developing this sleep rhythm. So they mostly wake up during day and then sleep during the night. And before any ActiWatch that people recording sleep and activity, it was hard and people have to manually record. But many years ago, people really developed this ActiWatch that basically you are wearing it and then you are going to detect your motion. So it's been used by multiple study. This is a tip example of ActiWatch was used for bipolar disorder patient monitoring. And you can see there, this is basically activity recording. And when you're sleeping, there's no activity. During day, you record the activity. The normal individual has pretty regular circadian rhythm of the activity. But bipolar or borderline personality disorder disease, they have much more irregular activity in their recording. And recently, even your smartphone can actually monitor circadian rhythm and then activity and then your sleep. And this very affordable and very compact size ActiWatch is available to apply for many patients, actually. Not only sleep-related disease, but shown here is an example from the spinal cord injury case. Sorry. For the disruption. So the circadian rhythm actually regulates almost all our physiological events, from our wake up and then sleep and bowel movement and blood pressure. So there is a time for everything. For example, the best coordination happens around in the afternoon time, and then high spike blood pressure happens in the evening time. And then in the midnight, there is a 2 a.m. deep sleep time. And then as you get up, the sharp rise of blood pressure, and then all other physiological activities regulated by circadian clock. Shown here is, so people and human is a good subject of studying circadian rhythm. Plant is good model system for circadian rhythm. But historically, circadian study was really made a big advance using Drosophila as a model system. And that was done by this giant named Seymour Bender. And I always think that this picture is so interesting because the way he looking at the fly is like is so lovely. He loves this. He loved this model system. And then he did a very interesting example with his graduate student, Ron Kanopka, that they used the mutagen, and then they treat this mutagen to Drosophila and measure their rhythm. So basically, they are recording this manually of the fly rhythm. Basically, fly has diurnal rhythm, and they are recording their activity. And from this mutagenesis screening, they identify aberrant circadian period mutant. One was arrhythmic, the other one has the short period, and the other one has longer circadian period. And that was really the seminal finding that you can actually manipulate circadian rhythm by mutating the core clock factors. And many years later, from three famous scientists in the circadian field, Ross Fish, Mike Young, and Jeff Ho, they cloned this initial fly mutant, period mutant, and then received the Nobel Prize very recently. And then after that, like a fly circadian rhythm really developed based on their finding, because now you have the handle that where to kind of start studying. And in mammalian system, my advisor, Joe Takahashi, was the pioneer to clone the first mammalian circadian clock gene from the ENU mutant screening. So you inject ENU, which is a strong mutagen, to mouse, and then generate G1 and G3 to screen through mouse circadian behavior. And from this screen, he identified circadian locomotor output, cycle, kaput. They try very hard to make this gene name clock. Kaput means in German is not working. So clock output is not working. So in our lab, we are using this mouse wheel running system a lot. So basically mouse is single caged in the wheel, and this wheel running is recorded by the computer by the wire. So from the day and night cycle, we can record their activity. Shown here is a typical wild-type mouse circadian activity. This is a LD cycle, the light cycle and dark cycle, and then double plotted. So you can see that mouse nocturnal animal during the light phase, they don't run, but during the nighttime, they start running. And this running is pretty addictive. They know what to do as soon as you put them in the wheel cage, and they really like enjoying and then running it. And first 2 weeks, they are in the LD condition, 12-hour light light and 12-hour dark cycle. And then as you can see from here, the light onset dictates their activity onset because light has the masking effect. But as soon as you put them on the constant darkness, they start showing their free will running behavior, which is governed by endogenous clock. And their free running period is actually shorter than 24 hours. The C57 Black 6 has about 23.5 hour circadian period. So it looks like they are getting up a little bit early every day, but it's because of it's plotted in 24 hours. But they have very precise activity onset time. And during the dark period, which is 12 hours, they continuously run. They take a break and continuously run. And their activity offset is a little variable. So we calculate activity onset. We based on the activity onset, we calculate circadian period. That's endogenous free running period because this period is happening without any external cue. So here is the clock Newton mice activity. As you can see from here, first 2 weeks LD entrainment, they entrain to light-dark cycle pretty well, but as soon as you turn off the light, they start showing very long period. Basically, it looks like they're getting up late every day because their clock period is way longer than 24 hours. They actually have 26-hour endogenous period. And toward the end of the cycle, they become almost arrhythmic because you can only lengthen so much and it become arrhythmic. And many years later, Joe crystallized this clock gene, and then it was the transcription factor, basic helix loop helix protein, heterodimerize another clock gene, which is BMA1, and regulate circadian transcription regulation. So in mammalian clock system, light around blue light wavelength around 460 nanometer is the light that actually shift our clock, which is indicating that nighttime phone and then computer with all this blue light is bad for shifting your clock. And then it goes to the eyes, and then retinal ganglion cell has the photoreceptor named melanopsin. This is not the visual photoreceptor. This is for the circadian entrainment receptor. And then melanopsin activation, and then goes through the optic neuron, and then it goes to the anterior hypothalamus, and then it is that SCN that we call suprachiasmatic nuclei, which we call the center of the clock. So this is the first site in the brain that received the circadian rhythm, and then it goes this circadian cycle of gene expression and then electric activity, and then this circadian cycle information is transmitted by neuronal and humoral connection throughout the whole body. So it's not only SCN cycling, but it's all peripheral tissue. They, under the SCN's guidance, they all go their tissue-specific circadian cycle. And then as an output, this cycle actually regulates almost all physiology. So I mentioned SCN, we believe that it's a site of master pacemaker, that it actually regulates circadian rhythm in the brain, and then also peripheral tissue. And then people knew that SCN is master pacemaker based on the lesion study. So early 70s, a lot of people did this kind of lesion study, basically just put electrode and then melt like local brain area, like a lot of nucleus area neurons, and see if it impacts mouse circadian behavior. So when they did this experiment with lesioning SCN, they can see that mouse become totally arrhythmic. So from this point, okay, people really believe that without SCN, there is no behavior circadian rhythm. So behavior study is very good for circadian study. And then also after Joe's cloning clock genes, and then following many other core clock gene cloning, we now know what's the component of core clock oscillator. So to study circadian rhythm in our labs, we do a lot of this time point, because these genes, core clock genes nature is they are cycling during one day. So you can have multiple time points during one cycle of circadian day, and then do the Western blotting and see the circadian character of it. But as field move forward, we need more molecular system that we can monitor circadian rhythm. So a very long time ago, I made this HER2-lock mice, which is luciferase fusion to the one of core clock gene period 2 in endogenously. So it can have the fusion protein that has HER2 and then luciferase. So slicing those mice SCN, we were able to record this 7 days rhythm of SCN oscillation. And then culturing many other tissues from these mice tell us that clock is pervasively everywhere, not only the brain, but many different peripheral tissue, including the tail. So with using these mice, we ask very simple question that does peripheral clock, we're gonna stop if SCN clock is not there anymore. So we go back to the classical lesion experiment and then lesion SCN by electrode and then melting the SCN neuron and then confirm the animal behavior by the wheel running. That you can see that this control sham mice has no altered behavior, but SCN lesion mice shows completely arrhythmic. So SCN is gone, so I cannot culture SCN anymore, but take out the peripheral tissue and then culture them. And to our surprise, the liver from the SCN lesion mice show pretty robust circadian rhythm. And then after 2 weeks later, when we change the media, which is synchronizing the circadian rhythm again, because unlike with the neuronal cell, peripheral tissue don't have that much coupling. So if you are doing ex vivo culture, they become desynchronized, and the media change serve as a synchronizer. If there is like a cell remaining healthy, there's a cell that they will not gonna regain the rhythmicity. But in here, we can get all the good rhythmicity after media change. That means SCN still has functional molecular clock component without SCN. Then the question is, then what's the difference between the intact and SCN lesion animal? We look more carefully and then we decide to look for circadian phase and amplitude and internal phase control. So circadian rhythm consists with this 3 components. It's very important that circadian period length is from peak to peak or trough to trough, and amplitude is from trough to peak, and phase. Phase is kind of the time point then that when the specific event happens. So you can count the phase, the time point of first peak or second peak, something like that. So when we compare this internal phase, so our mouse tissue has their internal phase coordination. So for example, lung comes peak, lung circadian rhythm comes peak most latest, and cornea peaks most earliest. So in individual color indicated by one animal is indicated by one color. You can see that this internal phase between animal is very conserved. But in SCN lesion, this internal phase control is all abolished. So there is no more coordinates between the rhythm, between the inter-organ communication. And then when you see the single recording, you can actually see that they are actually out of phase. So when you're combining this, the intact animals still have nice circadian rhythm, but SCN lesion animals, they are just everywhere. So we learned that without SCN clock, still peripheral tissue clock oscillate, but because of there is no synchronizer, they are kind of oscillating in a random way. And their integrity between the tissue rhythm was abolished. That was what we learned. And then I just want to share very briefly about what I realized very recently. So when I studied circadian rhythm, I was very fascinated that certain gene expression is regulated by clock and then cycling with a period of 1 day, basically rhythm. And then this physiological output underlying molecular mechanism is core clock genes cycle. This core clock cycle, and then also they use the same cis element to regulate the clock control gene cycling. So core clock cycle is transmitted to the core clock control gene cycling, and they are regulating physiology. But the reason why they are cycling is basically not to cycle the system. the system needs to maintain their homeostasis instead of going up and down. So these genes going up and down to make the system to get ready to not to cycle in terms of in meeting against this environmental change. So that's so far my core clock work. And then in circadian field, a lot of people are very fascinated about SCN and brain clock, and then brain clock regulation of behavior. But not many people study the role of circadian clock in PNS, so like peripheral sensory neurons. And I thought that was very good opportunity. And then I kind of get so interested about this PNS neuron and then PNS circadian rhythm. And then as I learn more, I understand there is a trigeminal ganglion, which is a PNS for the head, and then DRG for the rest of the body. And then curious about, do they have circadian clock? And then if they have circadian clock, what's the impact of circadian clock in the PNS system? So, and as I interested, I realized that there is a rhythm of pain, that pain called their people is a fifth vital sign, that a lot of pain has circadian rhythm, but looks like no one is studying it. So I decided to jump in and then look for pain model and see that circadian rhythm of pain exists and then how it's regulated. So first, very briefly, I will describe about my first work related with the CIPN. And second part of my talk, I will describe my recent work about circadian clock regulation of headache. So I collaborate with Dr. Hee Kim in MD Anderson and then Dr. Abdi to understand the circadian regulation of chemotherapy-induced peripheral neuropathic pain. So after the chemotherapy, the patient developed this peripheral neuropathic pain. So the mouse model for this pain is treating mice for long-term chemotherapy reagent. In this experiment, we used the paclitaxel, like the Taxol type of the chemotherapy reagent. So as you can see from here, as we treated the paclitaxel, the mechanical sensitivity threshold really decreased in the mouse. This is the rat model. So after you establish this chronic pain model, we decide to look at their circadian rhythm of pain response. And then when we measure this pain in every 6 hours during one circadian cycle, you can see that pain sensitivity, the threshold is much more higher during the nighttime than daytime. So there was an obvious circadian oscillation of pain sensitivity. And then we use Per2-Luck mice model and then take out the DRG neuron and then culture them and then monitor the circadian rhythm. And there was not that much difference between this L1 to L5 DRG circadian rhythm. And at the end of the 7 days, when we give media change, they resynchronize and show the robust circadian rhythm again. So we show the peripheral neuron DRG has the good circadian rhythm. And then we decide to look for if core clock protein expressed in the DRG neuron, and then both period and BMAL expressed in neuron, and then also satellite cells in the DRG. Then we decide to look for the transcriptome and then decide to compare this transcriptome between circadian time points. So we did both dosahon and DRG. And then shown here is just showing that this is ZT8 versus ZT20. So basically middle of day and then middle of night. And we compare transcriptome difference between this daytime and nighttime, and then showing this number of DRG. And then also we are comparing between the treatment. So vehicle versus the treatment and vehicle versus treatment in 2 time points. Interestingly, this we consider as a circadian DEG has strong overlap with the time dependent pain genes, because this is comparison between vehicle and paclitaxel. So this is a pain sensitive gene at ZT2 during the daytime. And this is a pain sensitive gene during the nighttime. Between these two, overlap is small. So it tells us that in pain response, CIPN response, the daytime and nighttime with different gene involved, and then but they both share strong about 30% share of the diurnal circadian gene. So it tells us that circadian regulation for this pain response is definitely exist. And then pathway analysis point out important pain pathway genes. So I was very happy with the first work, and then next thing was it's not that I was really looking for to study the headache, but we have very good collaborator that wanted to understand this new disease, cluster headache, that the headache and very, very severe headache, but the most striking, it has very sharp circadian window. The patient can predict that I'm gonna have headache in 15 minutes later. And this headache happens at the same time of the day for 1 month per 1 year. So it's circadianly regulated disease and also circannually, which is yearly regulated disease. But we have no idea about circannual yet. But Dr. Mark Burish is our school neurologist. And then he's a headache specialist and cluster headache specialist. And then team up with Chorong Han, my very strong postdoc who was taking the lead role for this project. And then we were able to start working on this disease more than 6 years now. And then finally, we kind of understand a little bit because this was really no man's disease, no man's area, that there was no gene, no mechanism or nothing. A lot of study was actually more advanced in migraine field. In circadian rhythm and migraine is well established that migraine patients have very strong circadian rhythm. But cluster headache is really not much studied at all. And actually, our group is the only group that study cluster headache in US. And there's a very strong circadian regulation of headache. And then showing here is the migraine and hypnic headache. This is another type of headache and cluster headache. And as you can see that they have their peak time very clearly. The migraine around the morning time, cluster headache around the evening time. And as we actually show from the rat model, there is a neuropathic pain very strongly in human, showing very strong circadian phenotype as a kind of example. So for the cluster headache, and this is a survey result from the patient. So patient from the about this much cluster number of cluster headache patients. So they're all cluster headache patients, and they're asked to compare their cluster headache pain level to other pain they experienced. So they say cluster headache pain is the most highest pain. Compared to labor pain and bone fracture, this is the #1 pain they experience in their life. So it's very, very excruciating pain. And showing here is a tattoo of the patient that he wants to describe the pain like this, that someone is like drilling his eyes. So one side of face location, and then this timing of headache is about 15 minutes to 3 hours. Very, very precise. And then it has all this feature of the headache response. And then, as I mentioned, they have very strong circadian regulation. So from many different countries, their peak time of the headache onset is always 2 a.m. and it's in all different time zones. So wherever time zone you are, they experience the highest pain at 2 a.m. And in headache, the trigeminal vascular system is regulating this headache response, and TG is consisting the major part consisting the trigeminal vascular system. And TG, as you guys probably know, that this is a pseudo-unipolar neuron, and then it receives the signal from the face, and then it transmits this signal to the trigeminal nucleus, and then signal is going to thalamus, hypothalamus, and cortex. So it's kind of in the middle between the receiving the signal and then transmitting signal to the brain. And then our first question is, does TG have circadian rhythm? So we take the TG from HER2-locked mice and then culture. This is ex vivo slice culture of TG. And then we also dissociate TG and then culture it. So both tells us that it has very strong circadian rhythm. And when we do the dispersed culture and neuronal marking with the GFP, we can see the single cell has very nice circadian oscillations. So yes, TG has circadian rhythm. And then we also look for core clock gene expression in TG neurons and glia, and they show very strong core localization, and then they have very strong expression. And then next question was how we can actually monitor headache in a circadian time point way. So we use this nitroglycerin-induced headache model, which is enodonor, and is very well-known vasodilator. So the reason why it became the headache model system is because of NTG was treated for the coronary artery heart patient disease when they have chest pain, when they need the blood vessel dilation to reduce the heart burden. People people treat NTG, and then because of vasodilator effect, the heart burden reduced. But all of these patients complain about their headache as a side effect. So scientists catch it, and then we use this NTG as a headache model and confirm that this is a good headache model by treating the headache medicine. Migraine medicine or cluster headache medicine relieve this pain actually. So this as a proven model, we use this model to monitor either bone fray using the foot or orofacial pain that you are poking the face and then measure the grimace score. And then after we established this bone fray method, what we performed in our lab was first measuring the pain signal molecule in headache, CGRP, and then we monitor the circadian rhythm of this headache response. So in the vehicle, they don't really show the circadian oscillation of pain sensitivity, but in the NTG injected mice, they are much more sensitive with the mechanical stress during the daytime than nighttime. And you can see the quantification here. So headache pain actually showed the nice circadian pain response. And then we use one of the clock gene mutant mice that has no circadian rhythm, two double knockout mice that has no circadian rhythm as a control mice to show that it's a clock dependent. Currently, we are using more TG or brain-specific knockout mice, but this work was done using whole body knockout mice. And when we are using per1 double knockout mice, in the NTG injected mice, there was no fluctuation for the pain threshold compared to wild type. So in this experiment, we show that clock is required to show this pain sensitivity of the headache. Next, we take out the TG and then do the circadian time course to understand that what's the TG transcriptome is regulated by circadian rhythm. And every 4 hours in the constant dark condition, we take out the TG and then perform bulk RNA sequencing. And you can see from this heat map, this is a time point from 0 to 24 hours. You can see that there are genes that are peaking in a lot of majority peaking in the middle of daytime. And when we compare these cycling genes of control and then NTG treated mice, so the normal mice and then the headache model mice, interestingly, about 135 genes have the overlap, but these control cycling genes not cycling at nighttime. The control cycling genes are not cycling in the NTG model. So NTG has very de novo NTG-induced cycling genes. And then you can see that from the heat map. And interestingly, the core clock genes that known to cycle, they don't show that much difference. Some other than some phase or expression amplitude change, they don't show the much difference between wild type and then the NTG treated mice. And these are the phase shift map in about that 135 shared genes that even if they are still cycling, their phase is significantly altered. And interestingly, all these headache susceptibility genes that are known to regulate headache and migraine has a much more stronger response to NTG compared to the core clock genes. That NTG model actually severely changed this headache susceptibility genes circadian response. And when we do the pathway analysis, the shared part is definitely rhythmic gene because we are doing cyclic gene analysis. And then a lot of vasculature change and then regulation of different cytokine and different headache molecule change was accumulated in the NTG group. And we take one more step to do more circadian analysis. Basically, we are grouping them between their peak phase. So from ZT0 to ZT, from the early morning. So this is the beginning of the day to the nighttime. We're grouping them according to their peak phase of cycling genes. And then you can see the different event is accumulating in different time points. And interestingly, NTG group, they have this enriched pathway for sensory perception of pain during the early morning time, which is kind of correspond to when mouse are more sensitive to pain. So we kind of have this evidence, molecular evidence that this highly sensitive morning time. The next thing, what we did was we take out the headache drug data, headache drug that used for any type of headache. We selected about 150 drugs and then went through the drug bank to identify this headache drug target and then compare that headache drug target with our circadian genes. And as a result, we were able to actually understand more practical treatment solution that how we can actually use this our circadian knowledge to treat patients in a better way by using the chronotherapy idea. So these are all cycling genes, and then they have different peak time point. And then we, based on their peak time point, we can actually treat their drug. So like this is opioid receptor, and then this is adenosine beta receptor. And there are many different targets of these drugs, and then they have very specific circadian phase of their peak time. For example, serotonin receptor HTR2A, which is a target for a migraine and cluster headache drug ergotamine. And then this time is peaking at around ZT6. So we can consider using this kind of map to apply patient for the chronotherapy idea. So I would like to thank my lab, and then also Dr. Mark Burish, my collaborator, and then Chorong, and my long-term collaborator, Jake Chen. I'd like to thank Hyun from Baylor, the Anderson Higi, and my collaborator in Japan. And these are the funding sources. Thanks for your attention.